Hi, grade 10s. I'm sure like me, you're feeling all sold out. Well, we're near the end now, okay? So you might remember at the beginning, I said that we are going to look at how molecules move into or out of cells at the end. It's at the beginning, near the beginning of the PowerPoint, but I just feel like it fits in better here. Let's just open here, share screen. All right, so from slide 12 in the PowerPoint, and it's on pages 42 to 44 in your textbook. So we have learned that the cell is surrounded by a membrane, cell membrane, plasma membrane, and that cell organelles, ER, nucleus, mitochondria, chloroplast, etc., they also have membranes made of lipid bilayers. There's the lipid bilayer, okay? Now, you might remember me taking a small piece of paper and poking little holes in it. So those membranes, they only allow certain small molecules to pass through them, and it also depends on their charge, all right? Because of that, membranes are said to be semi-permeable, differentially permeable, selectively permeable, or partially permeable. And molecules can move across these membranes by diffusion, by osmosis, and something new in grade 10 is called active transport, okay? Now, I found this table, which is quite a good summary. So if we are talking about the different types of membranes, okay, an impermeable membrane, if you take a plastic bag and you put water in it, nothing's going through. So cell walls which are lignified, like the cells making up wood, which we're going to talk about later, lignin makes them waterproof, so nothing will pass through them, okay? So they are said to be impermeable. All right, there's no pores and particles cannot pass through that lignified cell wall. Then you get the permeable membrane, okay? Permeable membranes have got large pores. Cellulose. So remember I said cellulose consists of lots of polysaccharide fibers, and there are large spaces between, they're crisscrossing, there are large spaces between them, and it's easier for, very easy for large molecules to pass through cell walls, right? Then you get semi-permeable, all the other words, differentially, partially, etc., or selectively permeable membranes. So they've got those little microscopic pores, me poking the holes in the paper, and small molecules can pass through, like water, carbon dioxide, and oxygen. All right, and we're talking now about cell membranes, like the tonoplast surrounding the vacuole and all the other organelles in the cell. All right, so I know that in grade 9, you will probably remember when we did that whole section on diffusion and osmosis, but I'm going to go through it again, and it's to remind you, and particularly for the new grade 10s, all right? So if you have any questions about this, please book an ASP with me and I will help you. All right, so what is diffusion? Okay, diffusion is the random movement of molecules. It could be through the air, through water, or through a solid, all right? So for example, if I sprayed deodorant here, those molecules are going to spread through this room, all right? If the doors were closed, the molecules of deodorant would spread until it was equilibrium, until they are equally spread out between the air molecules. But because there are two doors open here, it will never be to equilibrium, okay? Because the molecules are going to pass out as well. They will continue moving from a region of high concentration where I sprayed the deodorant out the door, okay? And eventually I won't be able to smell it anymore. Now, why do molecules move? They move because they've got kinetic energy, and that's what you've learned in science. And they move randomly, and they bump against each other, and they bump against the air particles, okay, the little deodorant molecules, and they move from a region of high concentration to low concentration, the rest of the room. And if the doors are open, out, okay? We call that a diffusion gradient, from a region of high concentration to low concentration. You know, I had a cup of tea just now. So I poured the water and then you add the tea bag. And if I just leave the tea bag to sit here, then the molecules from the tea will diffuse through that little membrane to the rest of the water, okay? And eventually, I will have tea. But how do I speed up diffusion? I stir it. The same as when I add sugar. If I put sugar in my tea, I'd have to wait quite a while for those sugar molecules to diffuse evenly through my tea so that I can drink it. But what do? I speed up the diffusion rate by stirring with a spoon. All right, so diffusion is a passive process. It happens, all right? If your mom is cooking something nice in the kitchen, like I don't know, piece of steak or whatever, or eggs, you are eventually going to smell the food that is cooking, because those molecules move from a region of high concentration to low concentration through the rest of the house, in your nose, and then you can smell. So there's no energy that is required. The molecules move randomly because they have kinetic energy themselves. All right. So we've talked about deodorant, cooking smells, even perfume. Someone walking down the corridor and they put too much perfume or aftershave on. Okay, you all know about it. And sugar dissolved in water, or bath bombs dissolved in water. Lots of examples. Okay, what factors affect the rate of diffusion? Well, I've mentioned the concentration gradient. If I spray more deodorant here, it's going to spread faster. If I forget to boil the water and I pour the water in my mug and I put a tea bag in, I'm going to wait a long time for the tea to diffuse into that cold water. But if I boil the water, put the tea bag in, I'm not going to have to wait so long. The temperature increases the rate of diffusion because the molecules will have more kinetic energy and they will move, randomly, Brownian motion, and bump into each other, and eventually the tea will be ready to drink. All right. And also the size of the molecules. So if the molecules are smaller, they will diffuse quicker than if the molecules are large, okay? Because, they're heavier. They don't move as easily. All right, so there is a sugar cube dissolved in a beaker of water. And if you just leave it, eventually, over a long time, the molecules will diffuse evenly throughout the water. So we say until equilibrium. And the reason why it's until equilibrium is because this is a closed system. There's no doors or windows open. Okay, the sugar water is in the beaker, it's not going anywhere. So it's until equilibrium. All right. I will upload the little animations as well. Okay. So here we have the same scenario. So if you put dye or sugar or salt in water, you're making a solution. If you put dye in water, these molecules have got kinetic energy, so they randomly move. Okay, and eventually they will be evenly distributed, and you'd have a solution. Okay, obviously if you stir it, like I said, then diffusion rate will be speeded up, or if you heat up the mixture, it will also be speeded up. All right, now osmosis. Oh, what I wanted to say was molecules that can diffuse. We talk about gases and solutes like sugar and salt and dye molecules and things like that. Okay, cooking smells, etc., perfume molecules, etc. But there's a special type of diffusion, and that's known as osmosis. So osmosis is the diffusion of water molecules from a region of high concentration to low concentration. So it's also down, not really down, but figuratively down, from a region of high concentration to a region of low concentration. So if you just ate a whole packet of salty chips, all right, what do you want? You want water. Okay, so generally water moves from a region of high concentration of water, where there's mostly water, to a region where there is a solution with salt or sugar dissolved in it. Okay, now in grade 10, we also talk about high water potential. Water potential, so pure water, has the highest water potential. And water potential refers to the tendency for water molecules to move out of a system, from where there's a higher water potential to where there's a lower water potential. Okay, so for diffusion, we use the words high to low concentration. In grade 9 for osmosis, we also said water moves from high to low concentration, but now we've introduced the term water potential. Water molecules move from a region of high water potential to a region of low water potential. And we're not going to say anything more there because it gets quite technical if you ever study this at university. All right, now at the top here, water, if I just pour water out of a glass, I'm just pouring it, okay? In order for water to move by osmosis, it has to be through a membrane, okay? When you're talking about diffusion, I spray deodorant here, it just spreads. There's no membranes here. If I put sugar in the tea, it just spreads, okay? But when we talk about plants, plants absorb salts from the ground. Yes, the salts will diffuse from the region of high concentration in the soil through the root hair cells and their membranes into the plant root. So it is passing through a membrane. Okay, so diffusion can happen, in a space or through a membrane and into the root, for example. But when we talk about osmosis, you can't just say that water being poured out of a cup is osmosis. No, the water molecules have to pass through a membrane, and it's not always until equilibrium. Think of it this way. If it's just rained, the ground is wet. The water moves from the soil by osmosis. It diffuses through the cell wall and cell membrane into the first cell. So now that cell has got a higher water potential. The water moves from this cell through the cell wall and membrane to the next cell. Now that one's got a higher water potential. So it keeps moving up and up through the plants until it reaches the leaves. So there's never equilibrium, because we know that plants transpire. They're continuously absorbing water. As long as there's water, goes up the plant into the leaves, and then they transpire. And if there's not enough water in the soil, then they close their stomata because they don't want to wilt. Okay, but if it's obviously too little water around, then they are going to wilt. All right, so at the bottom here, so pure water has the highest water potential. Those water molecules, they can move about by themselves. There's no other molecules to hinder their movement, okay? They've got the greatest kinetic energy, and the minute salt or sugars or something are added, then they're kind of bumping against those other molecules, and then the water potential isn't so high. All right, now there's a lovely table on page 43 of your textbook, Osmosis in Plants and Animal Cells, that you can have a look at. You just do not need to worry about the words isotonic, hypertonic, and hypotonic. Okay, but you can definitely look at the movement of water into and out of cells. All right, so talking about osmosis still. So you must watch the videos. It's nicer to see what happens in the video, okay, because you can see the movement of the molecules. All right, so what do we have here? We've got a pretend system. Imagine a beaker divided in two by a cell membrane, like dialysis tubing, okay? And on the one side is salt water, a solution. The salt is the solute and the water is the solvent, so it's a solution. And on the other side, there's pure water. So which side has the highest water potential? The pure water, because those molecules are not being bumped around by these very large salt molecules. The water molecules are small enough to pass through the membrane. The salt molecules are too large, all right? So what's happening here? The region of higher water potential or higher water concentration is in the pure water. The lower water concentration or lower water potential is in the salt water. But hang on, why are some of these arrows facing from left to right? Well, that's because water molecules, they're small enough they can pass through the membrane in both directions. But if you look carefully, you will see that more of the arrows, 1, 2, 3, 4, 5, 6, more of the arrows are facing towards the left. So more water molecules are moving to the salt water solution, and some of them are moving only a couple of them have moved into the pure water solution. So we talk about a net water movement, okay? So what's going to happen on this side? If more water molecules move to the salt water solution, the volume of this side is going to increase. If more water molecules move out of the side with pure water, this volume is going to decrease. But it's still pure water, there's just less of it. So the water concentration is still high, there's just less volume. On the other side, however, because more water molecules moved into the salt water, the volume goes up, but now the salt solution is becoming more dilute, okay? So the water concentration is increasing, okay? There's more water on the left-hand side now dissolving the same number of salt molecules, all right? So now the way they've drawn this is quite nice because it shows you these large salt molecules. They attract the water molecules. They hinder the movement of the water molecules, all right? And that is why this side has a lower water concentration and more water molecules are drawn towards the salt water solution. All right, and you can read through this on the left-hand side. Okay, there's another diagram. So you've always got to look at the diagrams carefully. What are they labeling? Okay, so what do we have here? At the beginning, there was that membrane again, same type of diagram, but it's showing the volume. All right, there's the sugar molecule dissolved in water, and here's pure water. So there's a net movement. The arrow is showing that there's a net movement of water molecules from a region of high water concentration or high water potential through the membrane to a region of low water concentration or low water potential in side A. So therefore, side A's volume goes up, side B's volume decreases. It's still water here, so the concentration is still the same. There's just less water, but because more water has moved to side A, the sugar solution becomes dilute, so the water concentration has increased. Okay, it's going to taste less sweet. All right. Now, I just found a few diagrams on the internet. You will see in the little videos I've made of how to show osmosis. You can watch those after this. So we could use potato tissue, and we could make little cylinders of potato, like when you make potato chips. So what have they done here? They've put original potato cylinders that were all the same size, controlled variable, and they put them into different solutions, the same volume of solutions in 4 beakers, another controlled variable, and they would have left them there for the same time, another controlled variable. The independent variable is what solutions the potato chips were placed in. So this potato chip was placed in pure water, all right? This potato chip was placed in a dilute sugar solution, all right? And this potato chip was placed in a concentrated sugar solution. All right, they say here that the experiment was left for 40 minutes. All right, so what will happen? There's the original size of the potato chip, and after 40 minutes, it increases in size. Now, why has that happened? Well, the potato cylinder is made of cells, and in those cells, there's water and salt. So the higher water potential or concentration is outside in the pure water in the beaker. So there will be a net movement of water molecules into the potato cells. The vacuoles will swell, push against cytoplasm, the cell walls will expand a bit. So now those cells are turgid, and the potato cylinder increases in size. Okay? What happens if you put the potato cylinder, that's the original size, if it was put into a dilute sugar solution? So maybe a teaspoon of sugar dissolved in the same volume of water. Well, now the potato cylinder shrinks. Why does it do that? Because the higher water concentration or potential is in the cells of the potato. In other words, they've got more salts and sugars than the sugar water outside. So there's a net movement of water out of the potato cylinder. And the vacuole shrink, and then therefore the cell walls, cave in a little bit, and the cells become flaccid. Okay, last one. So what happens if you put the potato cylinder in a very concentrated sugar solution? Pile the sugar in that solution. So now the higher water concentration is going to be in the potato cylinder, all right, still. But the solution on the outside is a very sugary, sweet solution, sugar solution. So lots of water is going to move out of that potato cylinder, and it's really going to shrink a lot. So we always talk about net movement of water, all right? If the potato cylinder was placed in a solution that is equal in concentration to the cell sap of its vacuoles, there would be no net movement of water. Water molecules would still move in and out of the potato cells, but there would be no change in size of the potato chip because the concentrations are the same. Right. This is another experiment that I set up in the lab. All right, so what do we have here? They've got water in a beaker. They've made a little dialysis tubing bag. You'll see how I do that in the video. It's also called whisking tubing. It acts like the cell membrane of a cell. You can make a make-believe cell. And then they put sucrose, sugar solution on the inside. So would water molecules move into and out of that little tubing bag? Yes, because water molecules are small enough to pass in both directions. But because there is a concentration gradient, more water molecules would move from a region of high water concentration or potential to a region of low water concentration or potential in the sugar solution. So that's going to cause the level of the liquid in the glass tube to rise. Okay, that's how we can demonstrate osmosis. Now, I will load those up as well. Okay, what about animal cells? So the example that's often used is red blood cells. Whoops, wrong way. Okay, so if our blood cells are moving around in our blood vessels, they're happy, and your body is always controlling the concentration of water and sugars and salts and everything in your blood, gases, carbon dioxide, etc. Okay, so in normal blood, there would be no concentration gradient. Okay, the concentration of substances on the outside in the blood plasma will be the same as inside the cell. So there's no net movement of water into or out of the cell. And your body's controlling that all of the time. Your kidneys get rid of excess salt and water. Everything's being controlled. Your lungs get rid of excess CO2. They're bringing oxygen, all of that, okay? So that's in normal blood. The blood cells will have this, they're dented in from both sides. They're said to be biconcave. They've got lots of surface area to absorb oxygen. All right, so here there is no net movement of water into or out of the cells. However, if you then place blood cells into a concentrated salt solution, all right, so now you can see how the colors become darker. All right, so now these water molecules will still move into and out of the blood cells, but now the concentrated salt solution is on the outside, and there's a higher water concentration on the inside of the cell. Okay, so higher water concentration inside the cells and a lower water concentration outside the cells in the blood plasma, because salt lowers the water concentration. There's more salt. So there's going to be a net movement out of the cell. So they look like this, the biconcave discs, but because they lose so much water, they actually become this sort of bumpy shape. And that's known as crenation. Okay, the cell membrane becomes very wavy. All right, so in other words, if you are very dehydrated, you haven't got enough water in your blood plasma, therefore that's what can happen to your blood cells if you are dehydrated and lost in the desert for days. All right. Now, what happens if we place red blood cells in pure water? Okay, so now the higher water concentration is outside compared to the water concentration or water potential on the inside of the cell. So there is a net movement of water into the cell. But blood cells are only surrounded by a cell membrane. So they become really swollen up. They are not going to fit through blood capillaries anymore. And eventually they could burst. All right. Now this could happen if you drink, crazy people on YouTube. They get challenged to drink like 2 or 3 liters of water in a half an hour. Well, you probably won't be able to do that. Okay. But if you do, you're putting your kidneys under so much pressure to try and get rid of all that excess water. And there's going to be too much water in your blood, and your heart's going to be stressed. You're going to have high blood pressure, trying to pump this greater volume of blood around because all the water goes into your blood and your blood cells could burst. So things like that, we shouldn't go there. All right, so here's another picture. Okay, so here are the words iso, etc., tonic. Okay, so isotonic means that the concentration of the solution outside and inside the cells is the same, so therefore there's no net movement, like isosceles triangle. The sides of the triangle are the same. Okay, hypertonic, so here it means hyper, lots of salts in the solution outside the blood cells, like hypermarket. Okay, there's lots of things that are sold at Pick and Pay hypermarket. So there's a net movement of water out of the cells to the more salty solution. Hypotonic, hypo means low, okay? So if the cells are placed in pure water, there's a hypotonic solution, so therefore more water molecules move into the cells and then they could swell up and burst. All right, now there's one more section that we have to discuss, something new, and that is called active transport. So diffusion and osmosis, it's the passive movement of molecules from a region of high to low concentration or high to low water potential. Okay, so if this is the lipid bilayer, molecules can move into and out of cells, but sometimes they need a little bit of help. Gases and water can simply diffuse into or out of the cells through the lipid bilayer. Glucose, amino acids, salts, and water can, either pass through by itself or it can be assisted. So that's called facilitated diffusion. If you remember, some of those membrane proteins in the lipid bilayer, they've got little channels through the middle of them, and they will help the molecules to move across the membrane. Okay, but both of these are passive. In other words, no energy is required. But then there's something called active transport. Okay, the passive transport of molecules is always from high concentration, whether it's a gas, or high water concentration if you're talking about water, from high to low. However, active transport requires energy. So think about this. You've just eaten a meal and carbohydrates are broken into little glucose molecules, and now they're in your small intestine. And they move, initially they move by diffusion by facilitated diffusion, they move into your blood vessels to be taken to the cells. But then there's just a few glucose molecules still left in your gut, and there's more in the blood. So now the concentration gradient has swapped. And normally we say that molecules diffuse from high to low concentration, but your body wants all the glucose, okay? So there's a low concentration of glucose still in your gut, your small intestine, and a high concentration in the blood capillaries. So what happens is there are mitochondria, all right, in the cell, and they provide energy. So those glucose molecules are pumped across the membrane against the concentration gradient from low concentration in the intestine through the membrane proteins to high concentration in the blood vessels. It's a way to make sure that your body can get all those glucose molecules, even if the concentration gradient has shifted. All right, I hope you understand that. Please book an ASP if you are getting a little bit confused. All right, so there's the summary of active transport. It's the movement of substances across a membrane from a region of lower to higher concentration against the concentration gradient, not the norm, okay? ATP is used because the mitochondria break down sugar to release energy, all right? The substances bind to those membrane proteins, which help carry those molecules across the membrane against the concentration gradient. And that's the example that we were discussing, the uptake of glucose from the intestine into the blood capillaries, or plants. If they still need, there's a few salts left in the soil, but they still need them, all right? So ATP energy is used to pump those salt molecules from a region of low concentration in the soil to high concentration in their cells against the concentration gradient. All right, and there's just a diagram. So, this is how it happens. The protein molecules, it's almost like a little gate. So they pick up the molecule and then they open up on the other side. Pick up, release, and that requires energy. Diffusion and osmosis do not require energy. It's a passive process. Okay, and that's the end of my story.